For this food microbiology video assignment, our project title is Tempeh fermentation. Food fermentation is a microbial process in which beneficial microorganisms convert sugars into acids, gases, or other compounds. This process plays an important role in food preservation, while also enhancing flavor, texture, nutritional value, and digestibility. In this project, our group focused on tempeh, a traditional fermented soybean product. Tempeh is produced through solid-state fermentation using mold from the Rhizophus species, which grow and bind the soybeans together into a firm, compact structure. The purpose of this video is to demonstrate the tempeh fermentation process, explain the microbial activity involved, and highlight the physical and biochemical changes that occur during fermentation.
First, rinse the soybeans thoroughly, place them in a blanch bowl and add enough water to fully cover the beans, at least twice their value. Soak the soybeans overnight for about 10 to 18 hours until they are fully hydrated. After soaking, rub the soybean together in water to loosen the husks. Remove and discard the floating husks. Repeat until most husks are removed. Next, drain the beans and transfer them to a pot with fresh water. Cook the soybean until they are tender but not mushy. Spread the beans in a thin layer on a clean towel or tray and allow them to air dry. Transfer the cooled beans into a clean, dry bowl. Add vinegar and mix well to help prevent unwanted microbial growth. Then sprinkle the tempeh starter culture evenly over the beans and mix thoroughly to ensure even distribution. Transfer the inoculated soybean into the Ziploc bags. Press the soybean into an even layer, not thicker than 3 cm to allow proper air circulation and temperature control. Then seal the Ziploc bag securely. Poke small holes in the Ziploc bag to allow proper air circulation. Place the packaged soybeans in the clean, well-ventilated environment at 27 to 33 degrees Celsius and allow them to ferment for 48 hours.
During tempeh fermentation, several physical, chemical, and sensory changes can be observed to monitor the progress of fermentation. The pH of soybean gradually decreases in the early stage due to the activity of natural occurring bacteria producing organic acid, creating conditions favorable for mold to grow. The sour smell indicates that the lactic acid bacteria are actively fermenting sugar and produce lactic acid. Color change. The color of soybean changes from yellowish to being covered with white cotton-like layer as the rhizophus mold grow and spread. In terms of texture, the soybean become tightly bound together as the mold mycelium penetrate and link the bean, forming a firm, compact cake. Light heat may be felt inside the tempeh due to metabolic activity, while gas formation is minimal compared to the yeast fermentation. These changes occur because rhizopus produce enzymes such as protease, lipase, and amylase that break down protein, fat, and carbohydrate into simpler compounds, enhancing digestibility, flavor, and nutritional value of the tempeh.
In conclusion, our group successfully produced tempeh with a firm texture, mild nutty aroma, and visible white mycelium, indicating proper growth of rhizopus mold and successful fermentation process. From a nutritional perspective, tempeh is rich in high-quality protein, dietary fiber, vitamins, and minerals. The fermentation process improves protein digestibility, reduces anti-nutritional factors, and contributes to better gut health, making tempeh a nutritious fermented food product. Through this project, our group learned that strict hygiene practice, accurate temperature control, and proper fermentation time are essential to prevent contamination and ensure consistent product quality. This project enhanced our understanding of the important role of microorganisms in food processing and preservation. For future recommendations, the use of standardized starter culture concentrations and monitoring parameters such as temperature, humidity, and pH could further improve consistency and reliability of the results. Additionally, conducting replicate trials and extending the fermentation observation period would provide more comprehensive data and help the process run more smoothly. Overall, tempeh fermentation demonstrates how traditional food practices can be improved through scientific control to produce safe, nutritious, and high-quality fermented foods. Lastly, thank you to everyone, especially lecturer, for the guidance throughout this project. Those who supported us directly or indirectly. We truly appreciate all the help and encouragement given. That's all. Thank you for watching.